What is photosynthesis? The word photosynthesis can be separated to make two smaller words: photo, which means light, synthesis, which means putting together. Plants need food, but they do not have to wait on people or animals to provide them. Most plants are able to make their own food whenever they need it. This is done using light, and the process is called photosynthesis. To make food, plants need carbon dioxide, water, and sunlight. Let's take a look at how these are collected by plants. Carbon dioxide from the air passes through small spores in the leaves that are called stomata. Water is absorbed by the roots and passes through vessels in the stem on its way to the leaves. Sunlight is absorbed by a green chemical in the leaves. Photosynthesis takes place in the leaves of plants. The leaves are made up of very small cells. Inside the cells are tiny structures called chloroplasts. Each chloroplast contains a green chemical called chlorophyll, which gives leaves their green color. Chlorophyll absorbs the sun's energy. It is this energy that is used to split water molecules into hydrogen and oxygen. Oxygen is released from the leaves into the atmosphere. Hydrogen and carbon dioxide are used to form glucose for food and for plants. Photosynthesis occurs in two stages, commonly known as light-dependent reactions and the Calvin cycle.
The light-dependent reactions are the ones that happen thanks to the energy provided by sunlight. These reactions happen in a part of the chloroplast called the thylakoid. More specifically, in the membrane of the thylakoid. In this membrane, there are proteins that perform distinct functions in the process. The two photosystems that are in blue in this diagram, containing chlorophyll P680 and P700 that receive sunlight and start or continue the electron transport chain. The hydrogen pump, in yellow, that as its name reveals, pumps hydrogen protons inside the thylakoid against the gradient with the energy provided by the excited electron. The electron acceptor, in purple, that forms NADPH, and the ATP synthase, in red, that forms ATP. We'll explain more of this in a second. The light-dependent reactions start as the light hits photosystem 2 and starts the electron transport chain. As the light excites the pigments in the photosystem, an electron jumps from chlorophyll P680 to initiate the electron transport chain. When the energy passes towards the inner part of the thylakoid, it breaks a molecule of water into O2 gas and hydrogen protons. The O2 gas is expelled from the plant into the atmosphere, and the hydrogen accumulates in the lumen of the thylakoid. As the electron transport chain continues from photosystem 2 to the hydrogen pump, this last one uses the energy provided from the electron to introduce hydrogens inside the thylakoid against the gradient. The electron transport chain continues from the hydrogen pump towards photosystem 1, where it gets recharged by a new surge of sunlight energy captured by the pigments in photosystem 1 and releasing it freshly charged from chlorophyll P700. The electron transport chain continues and goes to the electron acceptor, where it uses the energy to join molecules of NADP with hydrogen protons to form the electron carrier NADPH. The electron transport chain ends here. At the same time, the hydrogen protons that filled up the inside of the thylakoid need to balance the gradient, and to do so, some have to go to the outside of the thylakoid. The ATP synthase opens its channels and lets the hydrogens through. It uses the energy created by the difference in gradients to join molecules of ADP with phosphate groups in the stroma to form ATP. The products then are O2 gas, which goes out into the atmosphere, and NADPH and ATP, which go to play their part in the Calvin cycle.
Okay, so in the second part of photosynthesis, there will be no light needed in order to run the reaction. So this reaction is called the light-independent reaction, or the Calvin cycle. So imagine we have a beautiful plant here, and in one of its leaves, it has several plant cells. So in each plant cell, it has an organelle called the chloroplast, where photosynthesis takes place. And inside the chloroplast, there is a fluid that is called the stroma, which is this yellow thing. And here in the stroma is where the light-independent reaction will take place. So the main inputs for the reaction will be the ATP, plus the NADPH, which were obtained from the products of the light-dependent reaction, and the CO2, which is captured from the atmosphere in form of a gas. And the outputs will be G3P, which is the main output and, let's say, the ultimate goal for the Calvin cycle. So this image was retrieved from Bing Images, and in the first step of photosynthesis, we will have RUBP, which is ribulose 1,5-biphosphate, and the 5 means that each RUBP is a 5-carbon molecule with 2 phosphate groups. So it has 2 phosphate groups and 5 carbon, a 5-carbon chain. And so 6 RUBPs will be reacting with 6 molecules of CO2. And this is called carbon fixation. This means that the carbons that are taken from the CO2 are put into a solid structure, which is the RUBP. So this will generate 12 PGAL or 3 PGA, as you wish to call it, which is the phosphoglyceraldehyde or glyceraldehyde 3-phosphate, which is a 3-carbon molecule and a phosphate group. So energy has to be added in order for the reaction to happen. And that energy is going to come from 12 ATP and 12 NADPH. And when that energy is released, it will be released in form of 12 ADP and 12 NADP plus, sorry. So NADPH is a source of energy because of the excited state of the electron within the hydrogen. Okay, and as it goes to a lower energy state, it helps to drive the reaction. So this principle will be the same in the ATP for the phosphate group that is released from the ATP. So a phosphate is released from the ATP, which is in a high energy state. And again, it goes to a lower one, so it helps to drive the reaction. So from these 12 G3Ps will be created, which, as I mentioned before, this is the ultimate goal in the Calvin cycle. So each G3P is a 3-carbon chain with 1 phosphate group. But only from these 12 G3Ps, 10 will be used in order to run the reaction once again to recreate the RUBP. So in this part where 2 G3Ps are not used and are released from the cycle, these 2 G3Ps will be transported from the chloroplast to make glucose, or actually any kind of a sugar backbone, but mainly glucose. So this process with the 10 G3Ps that we have left, with the 10 G3Ps, we'll take again energy from 6 ATPs in order to run the reaction. So 6 ADPs will be released, and also 6 phosphate will be released. So in order for this in order to facilitate the entire Calvin cycle, there will be an enzyme that is called Rubisco that will help to run the reaction in an easier way, let's say. So Rubisco stands for ribulose 1,5-biphosphate carboxylase. So this enzyme will facilitate the entire Calvin cycle. And this image was taken from here, from Welch class.